For our next speaker for today's workshop, it is my pleasure to introduce Dr. Hagen Tilgner. He is an assistant professor of neuroscience at Weill Medical College of Cornell University, and he will tell us a lot more about single-cell Iso-Seq that I mentioned. His talk is entitled Single-Cell Isoform Analysis of the Nervous System.
So, hi, my name is Hagen Tilgner. I'm an assistant professor of neuroscience at Weill Cornell Medicine in New York City. I'm going to jump right in and give you a quick introduction about splicing. So consider a pre-messenger RNA, which is basically gibberish. And the process of splicing is sort of the process that will decide where exons are on this pre-messenger RNA, remove the introns in between, and come up with a final message. Now here, it was night in October, easily understood, but the cell may very well go back to a point where this intron was removed, realize that there was an exon in between, remove the two introns surrounding, and get a different message. It was night in the lonesome October. So if you've read Edgar Allan Poe, you may recognize this phrase from one of his poems. And that's one way to think about alternative splicing. Sort of similar messages, but with different connotations. But there are other ways to think about alternative splicing. So this is the FAST receptor, has been worked on for decades by now. And there are two isoforms that differ by a transmembrane domain. And when the transmembrane domain is included in the protein isoform, and this protein will sit on the cellular surface, and when the apoptosis signal comes, the cell will happily accept the signal and commit suicide. When this transmembrane domain is skipped, that will not happen, and the cell will simply ignore the apoptosis signal. So it's really a difference between life and death, and that's another way to think about alternative splicing. I'm going to talk to you about a couple of things here, and I'm going to start with how multiple variable sites on RNA molecules are combined into full-length molecules. Okay, there we wrote a couple of papers for that, and by variable sites, I mean TSS, alternative exons, and poly-A sites or RNA edits. And this first part was done during my postdoc work in Mike Snyder's lab, and it was joint work with a couple of other researchers, for example, with Donald Sharon and Fabian Grubert. There's another paper, PacBio long-read sequencing paper in 2013, which I didn't put here. And then with Fareshta Jahanbani, who was also in Mike Snyder's lab for linked-read sequencing. And then in my own lab, we started the single-cell long-read part, which was led by Ishan Gupta and Paul Collier. So consider these toy examples here. I think this is a problem. I do not think this is not a problem. Let's say we only have these two messages here. Then we have a semantic dependence. Okay, these two negations cancel each other out, and these two things mean relatively similar things. If we have only one of the negations, again, these phrases, I think this is not a problem, I do not think this is a problem, mean the same thing, but they mean the opposite of that. So that means we have a semantic dependence between the negations or exons. Now, semantics is sort of difficult to measure in genomics, but statistical dependencies are measurable, and that's one thing we started to do. So we looked at a variety of long-read sequencing technologies to do that. What you see here, every molecule is one read. You see two alternative exons here included in maybe 50 and 60% of the molecules. And what you see right away is that about 50% of the molecules include both exons. 30% include none, and then 20% make up the other two combinations. So you can calculate a p-value. You will find that this is a strong deviation from random expectation, meaning that these two exons sort of know what each other one is doing. And that means we have non-random pairing, a process that I call coordination, and that has been called by other people as well, coordination. Now, in a later paper, we estimated what the fraction of genes with multiple alternative exons that have such coordination events, and we came up to about 40%. Now, there's a pretty high 95% confidence interval around that. Why is that important? Well, that's the fundamental justification why we should do long-read sequencing. If all these numbers were here at 0, we should all be doing exclusively short-read sequencing, use that to calculate a probability for each TSS, alternative splice site, poly-A site, RNA edit, and so on, and then multiply up all these probabilities in order to get the probability of the full-length isoform. What that graph is telling you is that in about 40% of the genes, you would be wrong. So, and this was published in late 2017. There were other interesting papers too. Just right afterwards, there was a paper from the Netherlands, and when I say right afterwards, it actually means they did it simultaneously, where they came to similar results using different exon sets and different exact definitions, but roughly similar results. And then there was another paper in 2005 that is really a good read, where they focused on one gene and actually explained the mechanism how one alternative exon can be coordinated with another one. Okay, here's just another example that I want to show you. It's the gene MAPT, microtubule-associated protein tau. A very important neuroscience. There are many diseases that are associated to it. They're called tauopathies. And again, here you have one alternative exon and another one. You see that a lot of the molecules skip both exons. Some molecules include the first and skip the second exon. Just a very few skip the first and include the second, but what you rarely ever see is inclusion of both. Okay, so that means these two exons here are mutually exclusive. And that is another form of coordination. Okay, there's a summary. There's lots of papers that may be of interest, and of course there's some here that I was involved in. Again, joint work with Don Sharon and Fabian Grubert, and there's a couple that I wasn't involved in here. These are the long-read sequencing technologies. These are the coordination papers, and this is a thing that I haven't really talked about today, which is about the implications of coordination for proteins. Now, when I started my own lab at Weill Cornell, we started to think a lot about this beast here, and that means in reality very often we also think about mainly about this one here. Now, one very important thought is that when you measure, for example, DNA methylation or RNA isoforms or proteins, fundamentally what you're measuring is a linear combination coming from different cell types: neurons, ependymal cells, oligodendrocytes, astrocytes, microglia. I didn't put the blood vessels. All these cell types play very important roles, and they have different subtypes again. So we basically were looking to define a technology that could deal with this linear combination and assign long reads to the single cells and to the cell types from which they originated. And this is work that was led by Ishan Gupta and Paul Collier in the lab. And in a nutshell, what we do is we take bulk tissue, prepare a single cell suspension, and then use microfluidics. Here we use 10x Genomics, but you can use Dropseq or others. Run through the regular short read approach, which in a nutshell gives us a correspondence of barcodes to cell types. And that's what you see in this Tisney plot here. Every dot is one cell, and they've been grouped into cell types. Then we go into the long read sequencing, and we actually did PacBio and Nanopore, but focused most of the paper on the PacBio simply because the error rate was much, much lower. These are CCS reads, so there are very, very few errors in there. And then you get these long molecules. They have these short barcodes, and then informatically you will look for these barcodes in the list of previous barcodes, and therefore assign this molecule to this individual cell, which comes from a cluster of cells which has also already been labeled microglia. Then the other molecule here comes from this individual cell according to that barcode. Now this cell already looks like a neuron. Luckily, it comes from a cluster called Purkinje cells, which is a very important type of neuron in the cerebellum. So thereby we can assign reads to single cells, but also to clusters. And here, when you exploit this capacity of grouping single cells to clusters, we have here three types of neurons. I'm not going to go into details here, but there are cerebellar types of neurons. Every line is one molecule. The orange exons here are alternative exons. And you can see that the neuronal cells are characterized by two isoforms: inclusion of 1, 2, 3, 4, 5, 6 exons, or the isoform that just skips the fifth exon. When you go to non-neuronal cell types like astrocytes, microglia, OPCs, you rarely ever observe these isoforms, but you observe other isoforms. For example, the skipping of all of these exons, these alternative exons. So that means there's cell type specificity in the isoforms, at least much more here than there is at gene expression level, at least for this gene. And so you can go to iceformatlist.com. This is where we keep all that information. There is a section for the P1 cerebellum, which is this paper, and there will be a section for another following paper. What you see here are all the isoforms, and sometimes they will have gen code IDs, in which case they're known isoforms, and sometimes they will have reads, they will have IDs stemming from a long read, okay? And in that case, it was a novel isoform. And here you have one of these novel isoforms. Here you have the 10 cell types. And you see that it's the major isoform in astrocytes, OPCs, and microglia, but it's rarely ever expressed, and not at all actually, in Purkinje cells and two other types of cerebellar neurons. Okay, here we have another novel isoform, again, wasn't in the annotation. It's the major isoform in these three neuronal cell types, and it's rarely ever expressed anywhere else. Okay, and so we point out here isoforms that are novel with respect to GenCop, RefSeq, and UCSC. I just want to point out that we're really standing on the shoulders of these, and without the existence of these projects, it would have been very, very hard to deal with that. So the following is in progress, so you could do us a huge favor if you didn't scoop us. Right, so now this is about brain region specific isoform expression and how to explain it at a single cell or cell type basis. And it's on the bioRxiv and was led by a student called Anushka Joglika. So she focused on two brain regions, and I should say that there's many authors that contributed to that, so it's a long list if you look it up on the bioRxiv. So she focused on these two brain regions, PFC and hippocampus, and we focused at a time P7 when there's a lot of brain growth. There's more and more axons and dendrites that exist, and there's also a peak in gliogenesis. So it's a very important time in the development of the brain. We sequenced this quite deeply, so there were about 13 million bar-coded long reads, and they're all longer than 500 bases. There's quite some that are in this range of 800, 900. There's about 7.5, maybe 8 million that are longer than 1 kb, almost 5 million that are longer than 1.5 kb, 2.5 that are longer than 2 kb, and I think something like 2 million that are longer than 2.5 kb. And of course, this is only partially determined by the attributes of the machine that sequences them, and in large parts, but not entirely, actually determined by the distribution of the molecules that are being sequenced. And so the first thing Anushka did was to develop a gene-wise isoform testing procedure, and that actually allows you to have tests for differential splicing that are independent because there's only one test per gene. Okay, so if we have this hypothetical gene here with two alternative exons, and let's say we have these four fake reads here that we sequenced. In this case, we have all four combinations of these two exons are made. That means these are not coordinated. They're actually randomly paired, to use the vocabulary that I used in earlier parts of the talk. Now, in a short read world, what you would do is you would count each exon and count the reads that support the exons. For the blue, it's 2 here, and divide it by the total number of reads, which is 4. So you would get a PSI or psi of 0.5, or actually 50%. Yeah, so that gives you actually 2 inclusion reads, 2 exclusion reads, and those you can put in a 2x2 table, exon in, exon out. This has been actually done by many researchers. For example, the first RNA-seq papers that talked about splicing did that. Now the problem is that you now get all these tables, and there are often multiple tables per gene, so you have to correct for dependent tests, which is much harsher. So what Anushka then did instead was to take the number of reads for each isoform and divide by the total number of reads. So for example, for the isoform that includes both, you will get 1 read supporting it and 4 total reads, which gives you a 25% usage. We call that percent isoform values, pi, another Greek letter. And you can also count that as having support of 1 for this isoform, 1 for that isoform, 1 for that, and 1 for that. And so that can be translated into a table of n isoforms in 2 conditions as well. Could also do multiple conditions if you wanted to. And that can take into account TSS and polyA sites. Okay. Now the nice thing is now that we have exactly 1 test per gene, and thereby they are independent tests, and we can use the Benjamini-Hochberg correction. Now, once you have the concept of pi, you can define a delta pi for each isoform between 2 conditions, and you can now sum these up for a gene. Now, if you sum up all delta pi values for all the isoforms of a gene, then you get 0, because whenever one isoform goes up, another one has to go down in relative terms. If, however, you take only the strongest 2 pointing in the same direction, then you don't have this situation. And second, you make sure that you don't add up lots and lots of very small delta pi values. So that's exactly what we do. We say, well, we want to have a delta pi value of at least 0.1, or I should actually say 10%. And we ask for a significant p-value between two conditions. So you get this typical volcano plot just for isoforms now. And the nice thing is that when you do this isoform test for all these genes, you get 395 genes that have differential isoform abundance between PFC and hippocampus. When you do all the exon tests with a harsher correction for multiple testing for dependent tests, you only get 30. Now you may ask why we're 5 missing. That is probably because we're excluding some reads that are informative for exons, but are not completely full length. In which case for the isoform tests, they're not used. Now, most of the time, the isoforms that contribute to these differences between hippocampus and PFC for a gene are known isoforms, but there are exceptions. We found, I think, 76, somewhere close to 80 novel isoforms contributing to those. And since they were novel, we were wondering if they're really true. So we collaborated with a GenCode team at DBI, and they validated these 76 manually, and it turned out all 76 were validated. So we got 100% validation rate. We then asked what they actually did in terms of function. About half of them were protein coding. So it's really different protein isoforms being generated. Quite a lot were linked to NMD. So that means there's some difference in regulation of nonsense mediated decay between the two brain regions, intron retention, and just one was linked to a long non-coding RNA. Right. So now we have these isoform tests. The question is, can we actually understand whether the regulation or the difference between hippocampus and PFC comes from the TSS or the poly-A side? Well, we can, because the same test that we used for isoforms, we can also use for TSS and poly-A sides. And in 10% of the cases, we see that there's a difference at the poly-A side. About 27 where there's a difference at the TSS. Now, these may also harbor some differences in splicing within the gene, but if we only find them in the isoform test, not at the TSS, not at the poly-A side, we know that the difference actually came from splice site choice or differential exon inclusion. Okay, so that's the 64% here. Now we can address the question, which cell types underlie brain region-specific isoform expression. And it could be one specific cell type, for example, somatostatin-positive interneurons, or it could be multiple cell types, for example, neurons and astrocytes, or it could be no cell type at all, because what we measure at bulk could also be a difference in gene expression or cell type abundance in one of the two brain regions. So here I want to just give you an example. You see here hippocampal excitatory neurons. Again, every line is one molecule. You see this inclusion of this exon pretty high, maybe 80–90%. When you go to hippocampal astrocytes, it's still 50%. When you go to hippocampal OPCs, it's, I don't know, 70%. When you go through to the same 3 cell types in the PFC, you don't see inclusion of this exon. There's a little bit of intron retention of the previous one, and potentially they could be pre-messenger RNAs that would eventually give rise to the inclusion of that. But even if that were the case, there would be much less inclusion here in the PFC. So here we really have a case where multiple cell types show the same difference between hippocampus and PFC. In other words, brain region overrides cell type specificity. Now, how often does that actually happen? I'm not going to go into the details here. I just want to point out to you that it happens more than we expect by chance. So there's really some pressure for some genes to behave that way, but it does not represent the majority of cases. It's actually far from it. Now I want to show you another example, also via gene. Here we look at hippocampal astrocytes and PFC astrocytes. We see pretty high inclusion here of the 2 exons. When we go to hippocampal OPCs and PFC OPCs, the same, pretty high inclusion. And then when we go to excitatory neurons, we really see very high inclusion in this exon here, 81%, and then only about 23% in PFC excitatory neurons. So there's a difference, a delta pi of 58%. That's pretty drastic. And that means in this case, the difference that you observe when you do bulk sequencing between hippocampus and PFC comes from one specific cell type, excitatory neurons, where it's really drastic, and a lot of other cell types that actually don't show any difference. And altogether at bulk level, that makes a, let's say, small change, but nevertheless significant. That represents the majority of cases. Okay, so in summary, until here, we can say that the majority of cases, a single cell type explains brain region specific splicing. However, the case where brain region overrides cell type specificity clearly happens more than we expect by chance. So that there seems to be a program to enforce that for some genes. Now, the no cell type to blame at all, that's pretty rare. Okay, now we actually went to investigate isoform diversity in the hippocampus. And that links to brain region specificity because in the hippocampus, there are some cell types that don't have an equivalent in the prefrontal cortex. And so what Anushka did here is looked at some high-level cell types. So all cells are grouped into either vascular, myeloid, neuroglia, and neurons. And what she plots here is the percentage of genes that show differences in alternative splicing or TSS or poly-A site usage. And what you overall see is that whenever you compare neurons to some other cell type that's pretty high, it's much less in the other one. And here, when you subdivide these cell types into more, for example, the neurons are here subdivided into inhibitory neurons, excitatory neurons, granular neurons, and the neuroglia into astrocytes, RGL, oligodendrocyte lineage cells, choroid plexus cells. Then you again see a lot of red here in the neuronal to non-neuronal comparisons. Now, if you, however, compare only different types of neurons, okay, and here we didn't only use those 3 types, but we already split up inhibitory neurons into different types of inhibitory neurons, excitatory neurons into different types, and granuloblasts into different types. The neuronal comparison, usually you have this, I don't know, 8-9% of genes are show differences in isoform usage. There are some exceptions here, but the differences between non-neuronal cell types, so when you compare 2 types of non-neurons to each other, is on average larger, okay? But the big surprise came when we realized that within the non-neuronal comparisons, so when you subdivide these comparisons of 2 non-neuronal cell types into those that involve one specific cell types, choroid plexus epithelial cells, which are abbreviated as CPE here, so comparisons of CPEs to other cell types versus comparisons of 2 cell types that are both not CPE, there's a quite drastic difference. Even more surprisingly, we can trace now the part of the isoforms that matter. And there is a little bit going on in terms of splicing at the exons, a little bit at the poly-A site, but drastic differences at the TSS. And that has been really a big surprise to me because usually these programs feed into each other and one influences the other. But here it really seems that the TSS is a really drastic difference. So in summary, until here, we can say the strongest isoform differences in isoform abundance, or relative isoform abundance, I should say, occur when you compare neuronal versus non-neuronal cell types. Now, when you compare 2 non-neuronal cell types, for example, OPCs and astrocytes or choroid plexus epithelial cells, then you find higher differences than when you compare 2 neuronal cell types. Now, in this previous observation is strongly influenced by the CPEs, so the choroid plexus epithelial cells, which are in this region of the choroid plexus just next to the hippocampus. And this specific cell types, choroid plexus epithelial cells, show a drastic difference in the TSS choice more than in exons and poly-A sites. Okay, now I want to talk a little bit about functional consequences. So let me introduce you to one specific gene here, that's FGF13. It has 2 isoforms, the S and the VY isoform. Yellow on this plot means 100% expression in all of these cell types. Black means 0% expression. And what you see that in these excitatory neuron types, you have a lot of the S isoform being expressed. In inhibitory neurons, with the exception of inhibitory neuron type 3, which are Cajal-Retzius cells, you also see expression of the VY isoform. Now, and this is data generated by Susan Lin, a PhD student in Jeff Pitt's lab, and we're very thankful for her or for their collaboration. And it actually shows that these isoforms have dramatic difference in subcellular localization. Okay, so the S isoform is localized mostly in the nucleus, whereas the VY isoform is present pretty much throughout the cell and also in the processes, okay, which really strongly suggests different function of the 2 isoforms here. Now, again, we've made all this data available on iceformatlist.com, so you can check your gene. In the previous version, the old version, we had just 10 cell types and not that much sequencing data. Now we have 45 cell types, and there's so many cell types that it becomes very hard to see, but for many thousands of genes, you can download a PDF like this. There's different types of inhibitory neurons, different types of excitatory neurons, GNB, so granuloneuroblasts, then non-neuronal neuroglia, so that means astrocytes, OPCs, things like that, RGLs, then myeloid cells, so microglia and macrophages, and vascular cells. And so you can get this isoform expression for all of them. The color code gives you basically the relative amount. And so I'm going to just cut and paste a little bit here so you can see something. This is the gene CAMK2B. There's one isoform. It's an annotated, so known isoform. And in a type of PFC excitatory neurons, actually type 2, we have the major isoform is this isoform, which represents about 80% of the molecules in this cell type. But when you go to another type of excitatory neurons in the PFC, so both types of excitatory neurons have very fine resolution here, this isoform is only expressed at about 30 to 40% of molecules, and there's a lot of other isoforms coming up, including novel isoforms here and here. Okay, then in hippocampal interneurons, there's a very strong expression of a novel isoform, which becomes the major isoform, maybe 60%. And again, the fact that there's a read ID here, not an NSMOS-T ID, means that it's a novel isoform. Okay, until here, we can summarize that there's you can get 25 cell types quantified across these two brain regions. And yeah, if you are interested, then please go and check it out. We're very happy to hear what you think of the database. Now, we also developed a new approach, which we call spatial isoform sequencing, or slide isoform sequencing, short Iso-Seq, which basically uses 10x Visium technology with long-read sequencing. And what 10x Visium technology does, it basically assigns gene expression to every dot here. And what our technology does is it basically allows you to assign iceform expression to every single dot here. Now, this is a slice to the brain. This green area, this blue area, and this orange area together make up the cortex. This area here is the hippocampus, and the pink area here is the choroid plexus. There are many other areas that I'm not going to go into in the framework of this presentation. What we can do now is we can compare that to the single cell differences we see between the hippocampus and PFC. So remember, in the single cell data, we had separately sequenced hippocampus and PFC. And so for each exon, we can get a delta psi between those two regions from the single cell data. Now, in the spatial data, we can take this region here, which is the hippocampus, and compare to the entire region here. This is the cortex, which we can use now as a proxy for the prefrontal cortex. And we can get also delta psi for each exon that compares the hippocampus to the cortex. Okay, and as you can see, there's pretty strong concordance between the two technologies. So we think that this is really a good sign. Now, with this spatial technology, you can also show that the differential inclusion of exons. So here is CLTA, very famous gene. There's one exon here, which again, yellow means high inclusion, black means no inclusion, highly included in most of the hippocampus, not so much in the choroid plexus that I mentioned earlier. And then the second exon is actually pretty highly excluded all over the tissue, with some exceptions, for example, in the CA3 area of the hippocampus. Okay, and that is something we can also validate with our single cell approach. Now, another gene, PKM, high exclusion in the hippocampus, but skipping of in the choroid plexus of this exon. When you go to the second exon, it's pretty much skipped in the hippocampus, but included in the choroid plexus. So there's really a switch between these two. And just another gene, SNAP25, there's two isoforms called A and B, which are defined by two alternative exons. And you see this anterior to posterior gradient with lots of inclusion of the exon here and lots of skipping of the exon here, and the other exon going exactly vice versa. So in summary, spatial isoform sequencing with spatial gradients in isoform expression, and sometimes you find these precise exceptions to to global trends. That overall in the hippocampus there's a lot of skipping, but a couple of areas there's actually pretty high or some inclusion. So with that, I'd like to thank a lot of people. These are the people that are currently in the lab. These are lots of collaborators for my PhD times in Barcelona, my postdoc times in Stanford, and I mentioned papers with Fabian, Donald, and Foreste. People at PacBio and at Tilgner that have been helpful over the years, at while Cornell, cherished collaborators at Amsterdam and at Emory, previously at Stanford, and Ben Barris is also here. These are the authors of the Nature Biotech paper, which was led by Ishan Gupta and Paul Collier. The last work that I presented is now led by Anushka Jogalkar, and we have two collaborators that really work a lot with us, and Andre and Ahmed, that I'd like also to thank. Funding sources from our ones from the Brain Initiative and from NIGMS, and previously from the Leon Levy Foundation. This is where we're located on the Upper East Side, and so this is where postdocs get housing, and sometimes also senior research associates, so people that already have done a postdoc but like to continue. And we're actually hiring, so we're hiring both on the informatics side, which is partially data analysis and partially algorithms and software design to deal with these kind of data and other novel types of data that we're developing, and also in the wet lab, which concerns developing new assays, just like Iso-Seq was a new assay 2 years ago, and applying these to disease of the brain. And we're interested in a variety of diseases that affect the brain. So please come and talk to me or shoot me an email. You can shoot it here, hut2006@med.cornell.edu. We're very eager to talk to candidates.